And up next, thank you for your patience. We're having Dr. Eric Levine, who's a professor of neuroscience at the University of Connecticut.
Okay, so let me just get some confirmation that you can hear me, that you can see the slides full screen, and you can see this red laser pointer. So can you hear me and see the slides and see this red laser pointer?
Yes, we can.
Great, thank you.
So go ahead.
Thanks, Vanessa.
Eric, if you want to put it in a full screen mode though for us.
It should be. It's not showing that right now.
No, we're seeing your slide sorter.
Okay, let me try it again. Stop sharing. Try this one more time. Still not in full screen. Sorry.
If you go up to the top, it'll say slideshow. Click on that, and then over at the left, it'll say from beginning.
Working now?
Yeah, we still don't have it full screen. But if you look up at the top, kind of middle of the PowerPoint, it'll say slideshow. And then over on the left, it'll say from beginning once you click on that.
Okay, just did that.
Yeah, you're there. Just give that a click and you should be good to go.
How many monitors do you have up in your office? Do you have 2?
Yeah.
Switch where your main screen is, pull it over to the other one, and then, so go out of full screen, pull it over to the other one, and then go full screen. It should work.
Okay.
Or when you share, maybe share screen 2, if it gives you that option.
Yeah. Let me try that. I was trying to do it so that I could be looking at the camera and looking at the share at the same time. But sorry, I'll try this again now.
Perfect.
Okay, so now it's full screen, and now you can see the laser pointer. Yes.
Yep, go ahead.
Yeah. Okay, yeah, so I'll just have to look at the screen over here instead of the camera. Okay, thanks. So thanks for the opportunity to present this work to you. Sorry I can't be there in person this year. So the title of my talk is Identifying Genes That Contribute to Dup15q Syndrome Using a Human Stem Cell Model. So I'm not going to really review the clinical features of Dup15q syndrome, since I think this audience is pretty familiar with that. And I'm not going to review too much of the genetics, since we've had some really good talks already from Carolyn Shannon and Stormy and others. But just as a quick reminder, Dup15q syndrome results from maternal duplications, so duplications of the maternal allele of chromosome 15 from this Q11 to Q13 region of the chromosome. And these duplications can occur as a single duplication within the gene itself, which is an interstitial duplication, or as an extra piece of chromosome known as an isodicentric chromosome, which contains 2 extra copies of this 11 to 13 region. So when this region is duplicated, either interstitially or by the IDIC, there are many genes that are now going to be overexpressed. And so this includes UBE3A as well as these other genes here. And really the overall goal of our project is to try to understand what are the contributing roles of these different genes, ultimately to help identify therapeutic targets. But the work today that I'm going to talk about is going to focus on UBE3A, which we've heard a lot about. So the reason why we're going to focus on UBE3A is because UBE3A expression is imprinted. In other words, it's only expressed from one of your parental alleles. And in brain neurons, UBE3A is only expressed from the maternal allele, the copy that you inherited from your mother. On the paternal allele, there's this antisense transcript that silences UBE3A expression. So all of the UBE3A in our brain neurons comes from the maternal allele. Now, as I mentioned, Dup15q syndrome results from maternal duplication of this region. Paternal duplication usually does not produce Dup15q syndrome, may produce some mild autistic features, but does not produce the syndrome. So that suggests that UBE3A must play some kind of primary role because all of these other genes are going to be duplicated with either a paternal or a maternal duplication. So the thing that distinguishes the maternal duplication is the increase in UBE3A. And that's why when Stormy referred to UBE3A as most likely to be a driver, to play some kind of a central role in the syndrome. And what we really want to see is, what are the roles of these other genes as well? So the gene UBE3A actually encodes an enzyme, and that enzyme, its role in the cell is to target other proteins in the cell for breakdown or degradation. And so obviously if there's too much UBE3A in the cells, then there's going to be too little of some of the target proteins because there's going to be excessive UBE3A activity and you're going to get excessive breakdown or degradation of those target proteins. We don't really know specifically what those important target proteins are in brain neurons right now for UBE3A, but obviously that represents another potential therapeutic target that we'd like to investigate. And also it's worth mentioning again that loss of UBE3A, so a deletion or mutation of UBE3A on the maternal allele causes Angelman syndrome. So clearly too much or too little UBE3A is going to have severe neurodevelopmental consequences. So the role of these initial set of studies is to define the role of UBE3A in Dup15q pathophysiology. Before we can expand and try to understand the complementary roles of other genes, we want to figure out what is really UBE3A important for. So there's 3 aspects, sort of 3 components of my talk that I'm going to focus on here. So the first thing we want to do is we want to identify physiological features, or what we call phenotypes, in Dup15q brain cells and circuits. We want to find out what makes Dup15q brain neurons different. What are the physiological differences? Once we know that, we're going to ask 2 related questions. First, we want to determine if this increase in UBE3A expression is actually necessary for these phenotypes. Most likely, it's going to be necessary for quite a few things because we think, obviously, the reasons I described, it's a primary driver. But there may be some phenotypes that are actually independent of UBE3A that only involve other genes. And so that's going to be obviously important to know for developing therapeutics. So the way we're going to ask this question in the studies I'll show you is we're going to normalize UBE3A levels in Dup15q neurons, just normalize UBE3A, and see if that actually prevents the phenotypes, to see if UBE3A is actually necessary. The other aspect of the question is whether UBE3A overexpression is actually sufficient. So to address that question, we're going to overexpress UBE3A in unaffected neurons and see if the overexpression of UBE3A by itself is enough to mimic these cellular phenotypes that we've found. So that'll tell us whether UBE3A overexpression itself is sufficient, or maybe there is an important contributing role of other genes. Now, we're going to do these studies using our human stem cell model that I'm going to address in a minute. First, I want to just talk about the fact that we can't really do these studies using current animal models, right? Even though the animal models have the great advantage of being able to look at behavioral phenotypes. But the current animal models really only address the role of UBE3A overexpression. So those models are very important for determining what the specific role of UBE3A expression is. But in terms of determining whether it's necessary and sufficient, it doesn't really allow us to address the role of other genes. And in fact, some of the really exciting work that Matt and Matthias presented last night suggested that in fact, other genes may play important contributing roles to the establishment of phenotypes. So what we're going to do instead is we're going to use these stem cell derived neurons that I'll review again in the next slide, which derive from affected individuals with Dup15q. So the entire 15q11 to q13 duplication is conserved in these neurons. So these neurons are true models of the duplication of UBE3A in all the genes and allow us to ask these questions. And as I mentioned, identifying the roles of genes besides UBE3A is going to be very important. We want to know what genes are contributing to the disorder if we want to identify novel therapeutic targets. And also, as we start to discriminate the roles of other genes, we may be able to change the window of opportunity as it was for a therapeutic intervention. We know, for example, from the Angelman model mice that lose UBE3A, that restoration of UBE3A has to be done very early during critical developmental periods in order to rescue phenotypes. Some of these other genes that may be involved may also be important during development, but maybe play more contributing roles throughout life. And so they may be targets that could be addressed therapeutically at later stages during development or even in maturity. So that would be a really useful thing to discover. So our studies are going to use these human neurons that are generated from the induced pluripotent stem cell lines. We got a really nice introduction to that from Stormy's talk last night, so I'm not going to repeat all of that. But basically what this technology allows us to do is to generate human neurons that are genetically identical to the donor that they were derived from and study them at a cellular level, something that we've never been able to do with human neurons before. You know, living human neurons, living human brains, we can only study non-invasively. So this allows us technology to actually study these human neurons at a cellular level. And as Stormy went through, basically we use patient-derived skin samples or blood samples, expose them to reprogramming factors that turn them into induced pluripotent stem cells. These stem cells can then be differentiated into functioning neurons that we can grow in vitro. And these are some pictures of the different stages of this process. This is a colony of the induced pluripotent stem cells from an individual. They are then translated or differentiated into embryoid bodies. That's one of the differentiation approaches we use. Stormy mentioned there's actually a couple different differentiation strategies that we use these days. But eventually we derive these neurons that are grown in culture and we can look at their functional activity. So the first component of the talk is to identify Dup15q cellular phenotypes in these neurons. And before I talk about the isogenic line, I want to mention that the work that James Fink did when he was a student in my lab, he mentioned some of that in his talk. He compared the neurons that were derived from several different Dup15q individuals with several different unaffected controls. And through a lot of work, he was able to establish a number of really interesting cellular phenotypes that were specific to the Dup15q neurons. So we're building really on that platform now in the current studies. But in order to look at more subtle phenotypes, and especially if we want to start to perturb individual genes and look at how they contribute to it, we really want to eliminate any genetic variability between the different subjects that we're comparing. We really want to look at cell lines that are genetically identical, except for the genes that we're interested in. And so that's where we're going to use these isogenic cell lines. And Stormy also gave a great introduction to that last night to explain how this is done. They use genome editing using CRISPR to basically knock out this isodicentric extra chromosome. Stormy went through some of the details. I will just say, poof, look at that. Stormy uses CRISPR and they're gone. So Stormy and her team was able to take a line that was derived from an IDIC individual and completely eliminate that extra chromosome. So now we're just left with the normal paternal and maternal alleles with no increased UBE3A expression. And just to validate that this CRISPR correction did what we'd expect it to do, here's a profile of several of the genes that are found in the duplicated region, including UBE3A. You're looking at the level of messenger RNA, the level of gene expression for each of these genes. And you can see that compared to unaffected controls, the isodicentric dupe individual has elevated levels of most of these genes, and those elevated levels are corrected back down to baseline levels in the CRISPR-edited control line. So what that means is we now have neurons derived from the Dup15q isodicentric individual, and we can compare it to neurons that are genetically identical in every way, except they're missing that extra overexpressed piece of chromosome. So this is really a perfect platform for dissecting out the roles of different genes. And we're going to use these neurons to conduct electrophysiological recordings. I'll mention again at the end, but all of these recordings that I'm going to be showing you, all this data is collected by a current PhD student in my lab, Mara El-Amin. And Mara is actually in person there at the meeting. So you can get more details about her work from her if you get a chance to talk. So Mara conducted these electrophysiological recordings. So this is an example of a recording pipette that's patched on to an individual cell in the culture. And I'm going to show you data, 3 different aspects: intrinsic neuronal features of individual neurons, excitability of the action potentials that individual neurons fire, and then also changes in network synaptic activity. And so this is a subset of some of the phenotypes that James had previously shown, and we're going to eventually expand this out. But for today's talk, I'm going to focus on a few of these aspects. So the first aspect is looking at individual action potentials. As James mentioned, the action potential is what the neuron uses to communicate with other neurons in the circuit. It fires this action potential. One of the characteristic features of the action potential relevant to its function is its amplitude. We can measure the amplitude of these neurons. And so we recorded a group of neurons from the Dup15q neurons, and then a group from the corrected cell line that loses the entire duplication. And we did this at several different time points. First, I'm showing you an early time point. So this is just after 8 weeks of neuronal development in culture. And at that time, you can see that the amplitude of the Dup15q neurons is significantly higher than the amplitude of the corrected neurons. If we record later in development, so this is after 19 weeks, so quite a prolonged period of in vitro development. First of all, if you just look at the blue bars, you can see that the corrected neurons, which would be like neurotypical neurons, have increased their amplitude, which is what we expect. This is the maturation that we expect during development. They have a larger amplitude later in development. But the Dup15q neurons still have an elevated level even compared to that. So this phenotype of increased action potential amplitude is seen both early in development, is maintained even at later stages. Another aspect of the excitability of these neurons is the inward sodium current. It's a current that flows through the membrane carried by sodium ions. It's important for regulating the excitability of the neurons. We can record the size of this current also in Dup neurons and in corrected neurons. And similarly, at both the early and the late time points, there's a significant increase in the amplitude of the inward sodium current. Another important parameter for these action potentials is what's known as the threshold for firing. The threshold is this inflection point where the spike occurs, where you reach a critical activation of those voltage-gated sodium channels to create this spike. And as neurons mature, this threshold becomes more negative. It becomes a more negative number as neurons mature. And so if we look early and late, we can see that the Dup15q neurons have a more negative threshold than their corrected counterparts. So they're showing an earlier and a sustained greater maturation of action potential properties throughout development. We also wanted to look at the firing of these cells to determine if there was hyperexcitability. And so here's a train of action potentials in an individual corrected neuron after stimulating the neuron, and a train of action potentials in a Dup neuron. You can see the Dup neuron fires more. This is similar to the phenotype that James showed us in his previous talk, some of the data that he also collected when he was in the lab. And again, the same thing. So at early stages of development, the Dup neurons are firing at a significantly higher frequency than the corrected neurons. Over time, the corrected neurons do mature and they fire higher levels at 19 weeks, but the Dup neurons are still hyperexcitable. They're still firing at an even greater level than the corrected neurons. And the final feature I'm going to talk about is synaptic activity. And this is something that James also emphasized as being a very important parameter relevant to autistic features, as well as susceptibility to seizures, are both strongly affected by dysregulation of synaptic activity. So this is just a cartoon showing different neurons connected by their synapses. There are both structural and functional differences in synaptic activity in the Dup neurons. In Stormy's talk yesterday, she briefly showed some of the structural differences we see in the synaptic connections that can be corrected by UBE3A reinstatement. Here we're actually looking at the functional activity. So we're going to record the activity of an individual cell in the network and use it basically as a monitor for the amount of network activity that's going on. So in these traces, each downward deflection represents a synaptic event. It represents some other cell providing synaptic input to the cell we're recording. And this overall frequency of events is an indication of how excitable the network is. So at the early stages, these synaptic networks are not mature enough, so you really don't see any activity. We have to wait to the late time points. But at these late time points, you can see that the frequency of activity in the Dup neurons is much higher than the frequency in their isogenic corrected counterparts. So I'm going to summarize this first part by telling you what these neuronal phenotypes that we have seen so far in these isogenic cell lines. So we see changes in intrinsic neuronal features, which is the increased amplitude, increased sodium current, and the hyperpolarized firing threshold. When we look at the firing of these cells, we see that they're hyperexcitable. They fire at higher rates than the control neurons. And we also see an increase in network activity as monitored by this increase in excitatory synaptic transmission. And I think, as I mentioned before, both this hyperexcitability of firing and the increased network activity are both things that could predispose towards seizure susceptibility, as well as autistic features and cognitive deficits. And maybe one of the things that makes the seizures, for example, in Dup particularly hard to treat may be because there's sort of a multiple hits going on. So not only are the individual cells hyperexcitable, but the network is also showing increased transmission. And so a lot of times, if only a single component is affected, for example, in the network, there are compensatory mechanisms to make up for that. So if some cells are firing too much, there's usually some downregulation of the network to avoid things like seizures from happening. But in the case of Dup15q, maybe what's happening is that you're causing an increase in firing of the cell, but at the same time, the compensatory mechanisms are not working correctly, and you're also getting an increase in network activity, and those two things are sort of synergizing and producing inappropriate activity like seizure activity. So now we want to take these phenotypes and look at the role of UBE3A. So the first question we're asking is increased UBE3A expression necessary for these cellular phenotypes? So to ask whether UBE3A expression is necessary, we're going to use these antisense oligonucleotides that you've also heard a little bit about from Stormy as well as from James. And basically these ASOs, we can use them if they're targeted to UBE3A to cause a dose-dependent decrease in UBE3A levels. So what we're going to do is we're going to normalize UBE3A throughout development. So these Dup15q neurons have the whole set of overexpressed genes. We're going to specifically just normalize UBE3A throughout development and see if the cellular phenotypes are still produced. It's important that it's dose-dependent because we're going to use the UBE3A ASO at a concentration where we basically reduce it down to normal levels of UBE3A expression. We don't want to block all UBE3A expression because then we'd really be creating a model of Angelman syndrome, which we know is going to have its own set of complications. So this is validation from some work done in Stormy's lab, showing the level of UBE3A in the Dup15q neurons and the corrected level when we remove the entire isodicentric chromosome. That's what would be sort of the typical level of UBE3A expression in these neurons from a single copy off the maternal allele. And again, working with Stormy's lab, we were able to determine a concentration of this ASO that reduces Dup15q levels about to the typical corrected levels. So it's no longer elevated, but it's not decreased either. It's right about where it should be. And now we're going to use this treatment to see how it affects those cellular phenotypes. So I'm going to start here with the AP amplitude. Here's the data I showed you before. Here's the corrected level. The Dup15q neurons have a higher amplitude. If we prevent the increase in UBE3A expression using the ASO, again, throughout development, and then record the cells, we see that the increase in AP amplitude is non-existent, right? So it never produced that increase in amplitude because UBE3A was not expressed. And we see the same thing for the sodium current. We see the same thing for the threshold. And you see the same thing for this AP hyperexcitability. So in all of these cases, that cellular phenotype was prevented if we blocked the UBE3A overexpression. So this tells us that UBE3A overexpression was necessary in order to produce these phenotypes. It doesn't necessarily, there may be other genes involved as well, but if there's no UBE3A overexpression, then you don't see these phenotypes, so it is necessary. Interestingly, however, we got a slightly different result when we looked at the network synaptic activity. So when we look at network synaptic activity, the UBE3A ASO does cause a reduction in the frequency. So it's not as elevated as it is in the Dup neurons, but it's also not back down to baseline levels as it is in the corrected neurons. So this suggests that UBE3A is required in part for this phenotype, but provides some evidence that there may be other things that are contributing to this increase in network activity as well. And I'm going to come back to that now in this next set of studies. So we've shown that UBE3A is, the increase in UBE3A is required for most of those intrinsic phenotypes. We now want to ask the question, is increased UBE3A expression actually sufficient on its own to produce these cellular phenotypes? In other words, if you just increase UBE3A, will you see these phenotypes, or is there actually involvement of other genes that may be important as well? So to do this, we're actually going to take advantage again of antisense oligonucleotides, but instead of targeting UBE3A directly, we're going to target this ATS, this antisense transcript. This is the transcript that's responsible for silencing UBE3A expression from the paternal allele. So if we target ASOs against this antisense, we can unmask expression of UBE3A from the paternal allele. Now, in these particular experiments, we're actually using this paternal duplication line. Now, as you remember, the paternal duplication does not cause Dup15q syndrome. And actually, in this particular individual that this line is derived from, this person did not show any autistic features. But their paternal allele harbors 2 silenced copies of UBE3A. And the reason we use this line is because now when we treat with the ASO, we're going to get increased expression of both of these copies of UBE3A. So it's going to create a better model of the isodicentric UBE3A, the isodicentric Dup15q individual. So we use the ASO, we block the antisense transcript, and now we're going to have 2 extra copies of UBE3A being produced, which again will mimic the case with the IDIC15. And again, some validation from work done in Stormy's lab shows that here's the level of UBE3A in the path Dup by itself. When we add the ASO and we unmask, we produce an increase in gene expression of UBE3A that's similar to what we would see in an IDIC line. So now when we go back and look at those same phenotypes, I'm going to show you all 4 of these together. All 4 cases, increasing UBE3A was able to mimic those intrinsic neuronal phenotypes. So the increase in amplitude, sodium current, the hyperpolarized threshold, and the increase in excitability were all produced to some extent by unmasking UBE3A expression in these path Dup neurons. Now, I can't exactly say that UBE3A alone is sufficient to produce these phenotypes. The caveat is that we use the paternal Dup line so that we could get 2 copies of UBE3A. But the paternal Dup line also has 1 extra copy of all those other genes. So it's not exactly the same as the IDIC, which would have 2 extra copies, but it's not like an unaffected individual that wouldn't have those. So it looks like UBE3A overexpression certainly seems to play a strong role and can produce these phenotypes. But what we need to do next is actually increase UBE3A levels in a completely unaffected line, just increase UBE3A to see if it can still produce these phenotypes. But the other interesting thing is now if we go back to this network synaptic activity, increasing UBE3A, even in the path Dup that has the 1 extra copy of the other genes, is not sufficient to produce the increase in network activity. And as I mentioned, I think this increase in synaptic activity, James mentioned too, is probably a critical thing that's involved in both seizure susceptibility and development of autistic features. And this is strong evidence that other genes in addition to UBE3A also contribute to produce the phenotype that we saw in the Dup15q neurons. And so this is maybe somewhat in line with the lack of behavioral phenotype that is seen in the UBE3A overexpressing mouse that was presented yesterday by Matt and Matthias. So I'm going to give a summary now. So we showed that Dup15q neuronal phenotypes could be identified in the isogenic lines. This included the action potential maturation, hyperexcitability, and the increase in network activity. that UBE3A overexpression is necessary for the development of most of these phenotypes, and that UBE3 overexpression mimics some of these phenotypes, but is not sufficient to cause the increase in network activity. So in terms of ongoing work in future directions, one of the things we want to do now is with the UBE3A ASO, we showed that if you prevented any increase in UBE3A expression in those neurons, it prevented the phenotypes. But what we'd like to do for optimizing potential therapeutic interventions is to identify basically when and how long we need to normalize UBE3A in order to correct the cellular phenotypes. As I mentioned in Angelman models, it's been shown that really early correction of UBE3A is critical. In these Dup neurons, we don't know. So we want to really determine that can we treat late, correct UBE3A and still cause a correction of phenotype, or does it really also need to be very early? And that plays into our other goal of identifying the contributing roles of non-UBE3A genes and their time course for intervention. So using the same strategy that we used in these studies with an ASO targeted against UBE3A, we can use some of the ASOs that Stormy talked about yesterday that her lab has developed, targeting some of the other overexpressed genes in the region, and see if correcting those gene expressions can affect any of the phenotypes, and also importantly, see whether some of those genes may be playing continuing roles beyond just early development, so that they may represent therapeutic targets that could be attacked later in life as well. So in the long term, one of the things that we hope we can do by identifying the role of specific non-UBE3A genes is maybe inform the development of mouse models that could then incorporate these non-UBE3A genes. The mouse models, of course, are very important because in the mouse model, we can actually relate neuronal phenotypes to actual behavioral phenotypes. So that's really important. And you saw that the extensive work that went into generating those really incredible mice that were talked about last night that came out of Ben Philpott's lab. And Matt acknowledged that trying to go through gene by gene and to create other mice would be very labor intensive and may not really be worth it in terms of the cost benefit analysis. So using the IPS screening approach, we may be able to identify one or two critical genes that really contribute to the phenotypes, and those could be used in a much more targeted way to be incorporated into the mouse models and see what that does in terms of generating behavioral phenotypes. The other thing we'd like to do with this IPS stem cell platform is not only test ASOs against different genes, but just screen a variety of neural therapeutics using hopefully somewhat of a high throughput approach to use these specific cellular phenotypes and find out what drugs may be able to reverse those phenotypes, when and where they need to be applied, and again, ultimately then test those maybe in animal models as a prelude to being used as clinical interventions as well. So I want to acknowledge the people in my lab who contributed to this work. As I mentioned, Marwa Elamine has been critical, as this is really her thesis project, doing all these recordings. Tawana Robinson in my lab has also played a very important role in both our Dup and our Angelman projects. All of this work benefits greatly from our collaboration with Stormy and her team in terms of generating these isogenic lines, developing and validating ASOs, all really important work. And we also collaborate with Ionis Pharmaceuticals, who's helped in the development of some of these antisense oligonucleotides. And finally, I just want to acknowledge our funding. Both the Dup and Angelman projects that Stormy and I have worked on together have been supported by the NIH. I just recently received a 2-year grant from the Eagles Autism Foundation, specifically for this project to look at the role of non-UBE3A genes. And yes, that is the Eagles NFL team. The owner of the Eagles has a personal connection to autism, and a few years ago, he started this Eagles Autism Foundation to fund basic research into autism mechanisms. As mentioned, the Dup15q Alliance provided a graduate fellowship to James for the work that he did several years ago in my lab, and Marwa in my lab is currently supported by a graduate fellowship from the Schlumberger Foundation Faculty for the Future Award. And I'll stop there and take any questions. Thank you.
Oh, Matt's back on too.
Hi, Matt.
Hey, Eric.
I'm just going to do one question because we do have the crew from Roche on the other line waiting, and I know it's not a great time for them. So I'm going to take one, and then if you want to hang on after they're done, we can do Q&A too, just if that's okay with you guys.
Sure.
Yeah, because I have 3.
Just do one.
I'll do one.
Yeah, like one.
Okay. Do you think that you need to move forward with more research before you think that this would be picked up by industry to move into the therapeutic space?
Yeah, so that's a great question. I think one of the things that we can do that maybe industry may not jump on as readily is to use this approach to identify what other genes are important. And if we can provide some evidence for them that, for example, one of these GABA receptor subunits, for example, seems to play an important contributing role to cellular phenotypes, I think that's what's going to motivate industry to then think about developing therapeutics to that alone, to maybe combine that with UBE3A. So I think what we want to try to do is almost like a target validation for other genes that would then prompt industry to see what they could do with those targets.